Welcome back to Lymphatics, where we get to now talk more about the details of the cells and the primary and secondary organs that make up the lymphatic system. So, before we start to get into immunity and our innate and our adaptive immune systems, let's review what an antigen is. So our cells, on the surface of the cells, there are these proteins. And these proteins can be recognized as being familiar or being unfamiliar. For example, there are jerseys on a soccer team, and all of the team have the same color jerseys, so that way they can be recognized as the same team. So they're going to have these different shapes that can be recognized as familiar or unfamiliar. So your kidney cells and your liver cells and stomach cells, plus all of your cells, they will have these proteins called antigens that are on the surface of them. And our bodies will recognize them as familiar. However, viruses, bacteria, fungus, protozoans, and worms, and any other pathogen will have antigens on the surface of their cells that our body will recognize as foreign. And foreign antigens will trigger an immune response. And they are key to our adaptive immune responses and training our white blood cells or our immune cells how to recognize these foreign invaders and defeat them and get them out of our bodies. So for example, the reason when you get a bump when you get a mosquito bite is because mosquito saliva has antigens in it, and our body registers that antigen as foreign. So then our bodies will go and it will attack those antigens and it will cause that inflammation and irritation. So as mentioned on the last slide, antigens are like different colored jerseys in a soccer game. It helps your body know who is on your side, that's familiar, and who isn't, which it would be foreign. Another buzzword that we've heard in the cardiovascular unit is the term antibody. So let me emphasize, antigens and antibodies are not the same thing. Antigens are the protein markers on foreign and familiar cells. Antibodies are the weapons produced by our immune cells, specifically our adaptive immune response, that will bind to foreign antigens. And we'll discuss how this binding of antibodies to foreign antigens help fight an infection. Antibodies will not be discussed when we talk about the innate immune response, and they will exclusively be when we discuss the adaptive immune responses. So white blood cells are those cells that are immune cells, and they will carry out the adaptive immune responses. So these are the ones that will learn how to fight one specific invader or one specific bacteria or virus. And we've already learned a lot about lymphocytes in the cardiovascular system. And so we have three main types of lymphocytes. We have natural killer cells, which is on the next slide, and then we have B cells and T cells. So B cells and T cells are both made in the same place, and they will end up in the same place. but they're going to mature in different places in between their start and end points, which is how they earn their different names. The other cell type was the natural killer cell. And this cell, this is a lymphocyte that will go out and seek and destroy viruses and cells that are cancerous or infected with a virus. And when they find that cell that needs to be destroyed, they can release this enzyme that will actually puncture a hole in the cell membrane of their now enemy cell. If it's a bacteria cell, it'll puncture a hole in the bacteria cell membrane. And then it will release these granzymes, which are enzymes that can enter through that hole and start to degrade any cell enzymes that are inside of that cell. And since that cell now has no enzymes, it's going to begin to die. Then a macrophage can come right in and it can engulf and digest that dying cell. So we mentioned these immune cells within these lymphatic organs and lymph nodes. They are called immune cells and they're called lymph cells. They are derivatives of lymphocytes. And there are the three main types. So the natural killer cells are able to seek out and destroy the cancerous, virus-infected cells. They can seek out bacteria and they can detect transplanted cells. So even other human cells that do not belong to you, if you have that tissue transplanted, a lot of times you'll have to have an immunosuppressant. That way your natural killer cells don't go out and find that tissue and destroy it. And then the two main types that we're going to talk about, though, are going to be the T cells and the B cells. And those are going to, once again, get their name based on how they mature. The B cells, when they activate, they will proliferate, which means they will make a copy of themselves. And then they will turn into a plasma cell that can produce an antibody. And then the T lymphocytes, those are going to mature in the thymus, and they will receive an antigen from an antigen-presenting cell. We'll talk more about those in a couple slides. So the bone marrow has stem cells, and the stem cells will produce the different types of blood cells, including lymphocytes. All lymphocytes will start out in the bone marrow. B cells will stay in the bone marrow and they will mature and become immunocompetent in the bone marrow. So B cells stay in the bone marrow, which is how they get their name. And immunocompetent means it's the ability of the body to produce a normal immune response to an antigen. So for these B cells to become mature means that they can help produce that normal immune response to that antigen. They're basically getting trained. So during this maturation process, it's a 2 to 3 day process. The B cells are trained to react to a specific type of antigen. The B cells develop antibodies for that specific antigen. They're going to proliferate, making copies of themselves, and they're going to turn into another type of cell called a plasma cell. And those plasma cells are what can make the antibodies. T cells are first moved to mature in the thymus gland. And once again, that is located above the heart, right here in the superior mediastinum. So that is why these cells are called T cells, T for thymus. Maturation here is a process that also takes about 2 to 3 days. The T cells are going to be influenced by the hormone thymosin that's produced by the thymus. They mature and they become competent in the thymus, where they're going to be trained to react to a specific type of antigen. They do not develop antibodies like the B cells do, though. So you can think of it like this. The B in antibody, think of B cells. They are the body builders. So that's like a little memory hook that we can use to remember that the B cells are made in the bone marrow, and they will produce the antibody. So both B and T cells end up in the lymph nodes. Some also will go to the spleen. In the lymph nodes, though, they will circulate with tons of lymph fluid that will be filled with debris and potential pathogens. The B and T cells are going to be exposed to the antigens in those lymph nodes. Many T cells also circulate through the bloodstream, continually patrolling for antigens. And so remember that monocytes become macrophages. Monocytes are in the bloodstream, and they have the ability to leave the bloodstream and enter into the tissues. When they do that, they become macrophages. And macrophage means big eater. So they will gobble up debris and cancer cells and virus-infected cells, dead cells. They're just basically like a wandering garbage disposal. And they gobble up all of the bacteria and viruses. And when they do, they will engulf that bacteria or virus or whatever it is that they're eating through endocytosis, and it will fuse with a lysosome that has chemicals in it that will chop up that bacteria into little teeny tiny pieces. And then they will take one of those pieces from that pathogen and bring it to the cell surface, and they will display it on the surface of the cell. And they just wave it around like a flag, saying like, hey guys, here's a piece of this guy who's trying to invade, and I need you to go destroy it. It's like a wanted poster saying, this is the guy, let's go get him, we got to get rid of him. And so these antigens are something that is foreign to the body, and it will stimulate the immune system. And so because they actually present the antigen to other cells, they are referred to as antigen presenting cells, or APCs. So here is an image of a macrophage. These small rounded, or not rounded, small bluish rod-shaped structures. Those are the bacteria. And then this large purple cell is the macrophage. And you can see these projections of the cell membrane of the macrophage. Those are called pseudopods, which means false feet. And they're how these macrophages move around. And those pseudopods can also surround and engulf one of these bacteria cells, and it will bring that bacteria cell into the macrophage and it will start to chop up that bacteria inside the macrophage into little teeny tiny pieces. Some other lymphatic cells that are involved are called dendritic cells. Dendritic cells are antigen-presenting cells that are located throughout our epidermis, in our lymphatic organs, and mucous membranes. So they'll be located in our nose and throat and anywhere where there's an opening to the external environment. These cells will use a receptor-mediated endocytosis instead of phagocytosis, and they will present the antigen to a nearby lymph node. So they will basically intake this pathogen, and they'll be like, "Oh my gosh, now I'm infected. What do I do?" Okay, it's okay. I know where to go. And they will go to a nearby lymph node where there will be cells, macrophages, and lymphocytes ready to get rid of that infection. And then there are these reticular cells that will be found in lymphatic organs like those lymph nodes. And they are similar to the dendritic cells. However, they are going to stay put. They're going to be just in that lymphatic organ, and they will uptake any of that debris or bacteria or virus. And then they will say, "Oh my gosh, I am now infected. Hey guys, let's make sure no other cell gets infected with this." And will present the antigen to one of the other cells within the lymphatic organ. And when talking about lymphatic organs, people try to think about it like discrete organs, for example, the spleen or tonsils. But you have some things that are far less organized than discrete organs. You have what is called diffused lymphatic tissue. And remember that tissues are groups of similar cells. That's the definition of tissue. And so these cells are scattered. They're not clumped or clustered into groups. They're not gathered in a specific location or forming any kind of discrete organ. They are scattered lymphocytes throughout the body. They are present in any part of the body that is open to the outside world. So they will be in the respiratory system, the digestive system, urinary and reproductive systems, and they are just scattered white blood cells. All these things, like the trachea, the nose, the urethra, the lungs, anything that has an opening to the outside world will also produce mucus. And so remember that the job of mucus is to trap dirt, debris, and bacteria and not let those things into your body. The tissue that makes mucus is called mucosa, and it's a simple columnar epithelium or a pseudostratified epithelium that contains goblet cells, and these goblet cells secrete mucus. The lymphatic cells that we're talking about here are scattered throughout your mucosal. So they're called mucosa-associated lymphatic tissue or MALT. So now that we get a little more organized, we're going to have lymphatic nodules, sometimes referred to as follicles. So this is a dense mass now. We have lots of lymphocytes and macrophages, these cells that are starting to group together in response to a pathogen. And so this is going to be a more discrete feature. And this is a constant feature that we find in the lymph nodes, the tonsils, and the appendix. And we're also going to find these along the walls of the small intestines that are referred to as Peyer's patches. So now we have more clear anatomical and well-defined structure, and now it's starting to look like a real organ. And so we have two main types. We have a primary lymphatic organ, which is going to be where these cells are made and grow. And then we have secondary lymphatic organs, which will be where the mature cells will travel to and can be found. But these organs will be like other organs, and they will have a connective tissue capsule that surrounds the lymphatic tissue and separates it from the neighboring tissues. So the organs of the lymphatic system include the thymus, and the thymus is going to be located just right on top of the heart in that mediastinum. And then we have lots of lymph nodes that are going to be scattered throughout the entire body. We have our tonsils, which are going to be in the throat. There is the spleen that is located in the upper right quadrant behind the stomach. And then we have the bone marrow. So we talked about bone marrow in the cardiovascular system. We talked about it in hematology, and we talked about it with regards to erythropoietin. And so red bone marrow is involved in hemopoiesis, which is blood formation. And that means that's going to be involved in our white blood cell formation and immunity. So this tissue is soft, it's loosely organized, and it's highly vascular. We're going to find red bone marrow in children. Red bone marrow will fill up the entire diaphysis and the medullary cavity of a long bone, and it will also be found in the ends. But in adults, we don't find red marrow in the diaphysis in that medullary cavity. We have yellow marrow in that location. So in adults, we will find red marrow in the epiphysis or ends of the long bones, and we will also find it in our flat bones. And what's going to separate this red marrow from the bone tissue, the osseous tissue, is the endosteum. There are also going to be these cells called reticular cells that are located within that area, and they will secrete colony stimulating factor, or CSF, and that will tell these blood cells that they need to mature. As they mature, they will push their way through those reticular cells and those endothelial cells to enter into a sinus, and that sinus, once they enter into it, they will be allowed to flow away in the bloodstream. So this image shows the formed elements of blood squeezing through the endothelial cells and into the sinusoids, which cover or converge on the central longitudinal vein at the lower left here. So we have the central longitudinal vein, and then we have these sinusoids, and so you can see how they all converge together. And then here we have an adipose cell. I've got an artery, and you can see here there is a megakaryocyte, and those make platelets, and he is secreting those platelets into the sinusoid. Here you have a white blood cells squeezing through these these endothelial cells and getting into the sinusoid so it can go down into that central longitudinal vein. And then you have these kind of purplish cells. Those are the reticular cells. And so the reticular cells, those are the ones that will release the colony stimulating factor, telling those other cells that they need to mature. The spaces between the sinusoids are occupied by islands or cords of hemopoietic tissue, and this is composed of macrophages and blood cells in all stages of development. The macrophages will destroy malformed blood cells and the nuclei discarded by developing erythrocytes. As blood cells mature, they're going to push their way through those reticular and endothelial cells to enter into the sinusoid and then flow away. Here you can see a cross section. This is the head of a femur that has been cut through, and you can see where the red marrow converges with the yellow marrow. And here we have a cross section of red marrow again, and let's just go through it. So AD is adipose, MK is the megakaryocyte, the VS is the blood sinusoid, and HP is hematopoietic. So those are those red blood cells and those white blood cells that are developing. A discrete organ as the other primary organ is the thymus. So this is located over the heart. It is large in children and it is tiny in the elderly. It is a member of the endocrine system, the lymphatic, and the immune system. But it's a primary organ of the lymphatic because it will house the developing T lymphocytes. And it's also going to secrete hormones that will regulate their activity. It is bilobed and is located in the superior mediastinum, behind the sternum, and the aortic arch. And over time, this gland will shrink, and it's almost microscopic once we get into the elderly. And so this is the picture of the thymus on a cadaver. You can see here is the heart, and it is located right behind the sternum, just above the heart. So the thymus is a bilobed gland, and it will have a connective tissue surrounding it called the capsule. And that capsule will extend into the gland and it will start to divide the gland into lobules. And so that is called the septum, where the capsule starts to divide into lobules. And these lobules will contain lymphocytes that can turn into T lymphocytes. And so if you zoom in on one of these lobules, you will see lots and lots of lymphocytes. And those are the round purple cells. We will also have some of those dendritic cells, some of those antigen-presenting cells. But the thymus has a cortex and a medulla. The lymphocytes tend to be found in the cortex. Other cells, also like macrophages, will be found there. The thymus makes a hormone, thymosin, and the job of thymosin is to mature the cells into the T lymphocytes. And then those T lymphocytes will leave the thymus to provide immunity. So those were the primary organs. The secondary organs, we'll start with the lymph nodes. We talked about them a little already. They are located along those lymphatic vessels. They collect lymph fluid and will remove debris, pathogens like bacteria, before returning the supply to the subclavian veins. And so we're going to have some defensive cells, immune cells within these lymph nodes. We're going to have macrophages, which can engulf through phagocytosis and chop those foreign substances into itty bitty little tiny pieces. And then we have the lymphocytes that will provide an adaptive immune response to that specific antigen. Lymph nodes are widespread and they are very numerous. We have about 450 in atypical young adults, but they tend to be especially concentrated in the cervical, axillary, thoracic, abdominal, the intestinal, mesenteric, inguinal, and the popliteal regions. but they have two functions. They function to cleanse the lymph, and it's also the site of our T and B cell activation. It is an elongated bean-shaped structure, but it's usually less than 3 centimeters long, and it will have a hilum. So that's where we will have afferent and efferent arterioles enter and leave the medial portion of the organ. These lymph nodes will be enclosed with a fibrous capsule that have trabeculae that will further divide the interior into individual compartments. And the stroma of reticular fibers and reticular cells will be in that stroma. So here you can see the lymph enclosed with a fibrous capsule, and you can see that trabeculae that starts to partially divide the interior of the lymph node into compartments. The parenchyma is divided into the cortex and the medulla, and we're going to find the macrophages, these gobbling cells, as well as lymphocytes there. You can see the hilum, and you can see the efferent arteriole going there, and the artery and vein going into the node. So the parenchyma is divided into an outer C-shaped cortex that will encircle about 4/5 of that organ, and the inner medulla will extend to the surface at the hilum. The cortex will consist mainly of these ovoid to conical lymphoid nodules. And when the lymph node is fighting a pathogen, the B cells will multiply and they'll differentiate into plasma cells in germinal centers. The medulla will consist largely of a branching network of medullary cords that are composed of lymphocytes, plasma cells, macrophages, reticular cells, and reticular fibers. The cortex and the medulla will also contain lymph-filled sinuses that are continuous with the subscapular sinus. And then the lymph node will also act as a bottleneck. So I'm going to go back to a slide and I'm going to show you the afferent and efferent lymph vessels. So it's going to be different than the veins. And it's going to slow down the flow of lymph, and this will allow time for the cleansing of foreign matter. The macrophages and reticular cells of the sinuses will remove about 99% of the impurities before the lymph will even leave the node. If you look here, we have the afferent and efferent artery and vein that are going to come through the hilum. But when it comes to the lymphatic vessels, these are still in one direction. So you have the afferent lymphatic vessels, and we have many more afferent lymphatic vessels than we do efferent. So the afferent vessels are where lymph will enter into the lymph node, and then it will exit through the efferent. And I've only got one over here. And so what that's going to do is it's going to slow down the flow of lymph through the node, allowing that time for the macrophages and lymphocytes to clean up that debris. And it's going to create this bottleneck effect. So that way more fluid will go into the node than can leave out. So there are several afferent lymphatic vessels that will lead into the node along the convex surface of the node. And then lymph will flow from these vessels into the subscapular sinus. And it will percolate slowly through the sinuses of the cortex and the medulla, and it will leave the node through 1 to 3 efferent lymphatic vessels, and those will emerge from the hilum. No other lymphoid organs have afferent lymphatic vessels. Lymph nodes are the only organ that filter lymph as it flows along its course. And as the lymph travels through the medullary sinuses, the macrophages are there cleaning up debris. And with less ways for the lymph to leave the node, this forces the fluid to stay inside of that lymph node for a longer period of time. It's going to fill up these medullary sinuses, giving time for the macrophages and lymphocytes to clean up that fluid. Notice that there's some lymphocytes, but mostly macrophages there. And so your lymph nodes tend to be clustered. There are the cervical lymph nodes that are deep and superficial in the neck. Those monitor the lymph coming from your head and neck. Axillary are the lymph nodes that are concentrated in your armpit, and those receive lymph from the upper limbs and from the female breast tissue. Thoracic lymph nodes are in the thoracic cavity, and they are especially embedded within the mediastinum, and those receive lymph from the lungs, the airway, and the mediastinum itself. You have lymph nodes that are the abdominal lymph nodes that are located posteriorly in the abdominopelvic wall that monitors lymph from the urinary and reproductive systems. Your intestinal and mesenteric lymph nodes, those are found in the mesenteries. They are also adjacent to the appendix and intestines that monitor your lymph from the digestive tract. The inguinal lymph nodes are in the groin and receive lymph from the entire lower limb. And then there's the popliteal lymph nodes that occur in the back of the knee that receive lymph from the leg proper. So this is showing you areas of lymph node concentrations. I'd like you to take note of the superior mesenteric lymph node and the inferior mesenteric lymph node. So that should sound very similar to the arteries and veins we learned, because the stuff just doesn't go away. But these are those enteric lymph nodes that will collect from your intestines. And then this is going to show you the real thing, somebody's groin area. But see how small these are? And they just kind of really look like a grain of rice, about 3 centimeters. They can be really small. And take note of them in comparison to the femoral vein and artery that are noted for you. And sometimes there are some things that go wrong with the lymph nodes. Anything that goes wrong with the lymph nodes is called lymphadenopathy. And if those lymph nodes happen to be swollen and painful from responding to fighting a foreign antigen, that's called lymphadenitis. And lymphoma is cancer of the lymph nodes. And so when lymph nodes become cancerous, that can be very dangerous because of the anatomy of the lymphatic system. Remember that metastasis is when cancerous cells break free from the original tumor, and they will travel to other sites in the body, and they can establish a new tumor in that secondary location. Well, metastasizing cells can easily enter into these lymphatic vessels, and they tend to lodge in the first lymph node that they encounter. And then they're going to multiply there, and they'll eventually destroy that node. And this presents very similar to inflammation of the lymph nodes, except it's usually painless. And so the cancer will typically spread downstream to the next lymph node. And when there is breast cancer, because there is such an extensive lymphatic network in that breast tissue, typically treatment of breast cancer is a lumpectomy, a mastectomy, along with removal of nearby axillary nodes. It's a very intense and aggressive treatment because of the lymphatic system. So the next secondary organ that we'll talk about are the tonsils. These are patches of lymphatic tissue that are located at the entrance to the pharynx. And you actually have more tonsils than you can see, but these guard against ingested or inhaled pathogens. They are lined with a layer of epithelial tissue, and they're going to have these deep pits, these tonsillar pits that are lined with lymphatic nodules, and bacteria can wedge and grow and get deep into those pits, which are sometimes why they get swollen and enlarged. And so you actually have 3 sets of tonsils. You have the adenoids. Those get typically are what get removed. But if we were to zoom in here and look at the histology of it, just take a look at here. I have a capsule, and look at these deep crypts or pits. Well, those can get filled up with saliva and debris, which is one of the reasons why those tonsils get kind of gross and swollen. So you have 3 main sets of tonsils. You have the palatine tonsils, which are a pair of tonsils that are located in the back of your oral cavity. They are the largest ones and most often infected. And then you have the lingual tonsils, which if you see lingual, you should think of tongue. Those are also a pair of tonsils embedded at the sides of the root of your tongue. And then there's the pharyngeal tonsils, which are also known as the adenoids. Those are, that's a single tonsil. It's not a pair like the other two. That's on the wall of the nasopharynx. And here you can just see some pictures. I have a normal tonsils here, and these are 2 images of tonsillitis. It's caused usually by bacteria, and a tonsillectomy will be needed to remove the infected tonsils. The next secondary organ we'll talk about is the spleen. The spleen is located in the upper left quadrant of the abdominal cavity. There is 2 types of tissue. There is white tissue and red tissue, or white pulp and red pulp, I should say. The white pulp is named for the appearance in a fresh spleen. So when you're looking at that histology image down below, that is purple or violet in color because of the histostain that has an affinity for cell nuclei. So the white pulp appears in that purple, but the red pulp does still appear kind of reddish. That red pulp is filled with red blood cells, and the white pulp is filled with white blood cells. So that's where we're going to find the lymphocytes and macrophages is in that white pulp. And here we can also see the spleen located in a cadaver. You can see the spleen right here. And so this is right behind the stomach, and we have the 2 kidneys there. So right there. Your spleen is kind of like a big bloody sponge. So when blood goes through the spleen, the healthy red blood cells can come and go, but once they start getting older and more fragile, then they're not going to leave the spleen. Then the spleen becomes the erythrocyte graveyard. And there are lots of macrophages that are in the spleen that will break down these old and fragile red blood cells. In the fetus, the spleen is actually the primary site of red blood cell production until the red marrow is developed in the fetus. So in the fetus, we'll find red marrow in our flat bones, in the irregular bones, and in the epiphysis of the long bones, but also in the diaphysis. The diaphysis doesn't fill with the yellow marrow until after the child has grown. And so what can happen is actually what's called these splenic contractions. And so the spleen can squeeze or contract, and when it does that, it will add more blood to the bloodstream, and that can stabilize the blood volume through this splenic contraction. But it is very highly vascular and it's highly vulnerable to trauma and infection. And so you can actually live without it, and the liver can take up a lot of the functions. However, you will be much more prone to infection. If the spleen is ruptured, it's because it's so vascular, the patient can bleed out. And so if it is ruptured, a splenectomy may be required, and that will leave the person susceptible to future infections. Here we have just a picture of that histology where you can see the red pulp and you can see the white pulp a little bit better here. And then just like the tonsils, we have what's called Peyer's patches. And they are basically the same thing, but they are found in the walls and embedded in the walls of the small intestines. But they are just like the tonsils, and they look just like the tonsils. So sometimes they're going to be called a lymphatic nodule, but when it's in the small intestines, it's referred to as a Peyer patch. And so this is actually a really cool pic here. And what looks like a corn or a pine cone is actually the Peyer patch inside of the intestinal wall. And so we're going to end that here, and we will pick up next time with the body defenses.